Okay. Sorry for that delay. I was remembering to post that. Hi, I need to bring the exams again. Okay. So most people have gotten their exams. Those who haven't can grab a mid-recitation tomorrow. I'll make sure Dr. Maxwell has them. Okay, let's continue with signal transduction. Maybe? Oh, it would be good if I plug this in. Okay, so for those who have not heard the news, I have to remember to post it on Blackboard for people who don't come to class. Should I not?
Don't do it.
That'd be me. Exam 2 is changed to 1028. Those of you who have not heard that yet, now you've heard it. Those of you in TV land or wherever you are, hopefully you'll see it on Blackboard. If I remember to post it, someone email me say, hey, tell her to class and doesn't come to lecture, but the exam's got on Monday. Okay, yeah. I will address the muscle contraction question from the other day shortly. I got flooded with answers, which was beautiful, and so y'all get points. And I haven't put the points in yet, but they'll come, don't worry. Okay, so far we have talked about signaling, just the generic idea of what signaling is, some example pathways. We're starting from the top, so we started with extracellular signaling molecules. Then we've been talking about the types of receptors, and on Monday we were doing GPCRs. So this is what came up when we were talking a little bit about GPCRs and intracellular mediators and calcium release. And Tyler, who's not here, asked a question about if this is what happens with muscle contraction. I was like, well, no, but yes, but no, yes. And then that was a subject teacher question. And most of you guys did relate the fact that, yes, there's signaling that does result in release of calcium from the sarcoplasmic reticulum, which regulates muscle contraction through regulation of the troponin complex. Something like that, yes? You said that, you got points. And I think what threw me off in answering that question was here I'm talking about the ER, and then there was the sarcoplasmic reticulum. That's okay. So this pathway specifically is just another pathway involved with GPCRs where you're activating the G protein. It's a different kind of G protein, and it's activating phospholipase C beta. And then that cleaves PIP2, which results in diacylglycerol stays in the membrane, and IP3 goes and opens up the calcium channel in the ER and releases calcium into the cytoplasm. And that activates enzymes such as PKC, protein kinase C. And that further activates signaling. So CAMP, cyclic AMP, and calcium are two examples of intracellular mediators that activate other enzymes. I'm being purposefully vague here because I want you to get the basic idea. That activate other enzymes that relay messages and affect gene expression and or other effects on cell function. So you've got receptor resulting in either a release of cyclic AMP or calcium, and that activates other enzymes that affects gene expression or other cellular processes. Okay, so this is where we ended. We are talking about receptor desensitization and specifically about GPCRs. So we already spoke about receptor sequestration and receptor downregulation as ways to turn off signaling. And then there are also other ways, and most of them involve inhibition in some way of the signaling molecule. So here you'll have your cytoplasmic tail that's inactivated. Here it'll inhibit some other signaling protein. And here there might be production of an inhibitor that's going to inhibit signaling. So with GPCRs, here's one mechanism where you have phosphorylation of your GPCR. So actual activation of your receptor is going to activate a kinase called G protein linked receptor kinase, GRK, and that's going to phosphorylate the cytoplasmic domain of your GPCR. And then that's going to recruit, so that's going to create a docking site for arrestin, which will prevent signaling by preventing interaction and activation with the G protein. And, well, let's actually talk about what else it might be doing. So right here, I'm showing you a receptor that's been phosphorylated on its cytoplasmic tail, the cytoplasmic side. And then you get recruitment of this arrestin protein. What might this be doing? So if you think back to this, these different mechanisms for desensitization. So what might arrestin be doing? So there's two ways that it's desensitizing or inhibiting signaling. So there's one way that I just told you, which is it's preventing interaction with your G protein, right? So that's how G proteins work, where you have the activation of your G protein after ligand binds to receptor, and that goes and activates something else, which starts your signaling, right? And then there's this other thing that could be happening here, right, where you've got arrestin binding, and what else might it be doing? With another common mechanism of receptor downregulation that we've spoken about before, perhaps when we were talking about movement of proteins throughout the cell a few weeks ago. Okay, sequestration. Right. So sequestration. So what do you think arrestin might be doing here? What might it be binding to? Yeah. Is it what?
Is it covering the phosphate?
So it is covering the phosphate, but it's actually recruited by the phosphate. So rather than covering, because that phosphorylation is not activating, that's an inactivating phosphorylation, and it's creating a docking site for arrestin. So arrestin binding is dependent upon phosphorylation. So yeah, so it is so phosphorylation recruits arrestin, but what do you think it might be doing? Yes.
So it's blocking the cytoplasmic tail.
Well, right. So it's blocking the cytoplasmic tail. So that's what I just said. So what else might it be doing that results in sequestration? Yes.
Does it recruit clathrin?
Does it recruit clathrin? Maybe. Yes. It's exactly what I was looking for. That's beautiful. Thank you. Yeah. So it's kind of acting like adaptin and recruiting receptors into clathrin-coated pits. And so you get endocytosis, and that's how you get that sequestration. Yeah. So there's two mechanisms here that I'm telling you about of GPCR desensitization. One is just inhibiting interaction or activation of the G protein. Because that happens between the receptor and the G protein, that's, let's say. Say normally you'd have a receptor, your GPCR, and your G protein would be here. And that cytoplasmic tail activates your G protein. It's involved with activation of your G protein. So that's one way. So if you cover that up, then you won't get activation of your G protein. That stops signaling. But also, by recruiting clathrin and then endocytosis of your receptors, that's also gonna desensitize the cell. Okay? How do we feel about that? Does everyone feel happy? Okay. So, what would be the effect of a mutated arrestin? What if arrestin couldn't bind those phosphates? What would be the effect on signaling? It would keep signaling, so you wouldn't be able to desensitize, right? You wouldn't be able to downregulate signaling. Yes.
But like, if the phosphate, so does that mean the phosphate groups aren't binding to the?
So that would be if let's say, so remember, arrestin's a protein, right? And proteins have amino acids that are going to interact with those phosphate groups, and that's how it's going to bind. So the mutation I'm giving you to think about is a mutation that prevents interaction with those phosphates. So you're still going to get GRK activity, so you'll still get phosphorylation, but you won't be able to recruit arrestin. Okay? Does that make sense? Okay. Yeah. So the question I'm giving you to think about is what would happen if arrestin was mutated so it could no longer interact with those phosphate groups on the GPCR?
Oh, I just meant to, like, if the GPCR is phosphorylated, it activates the GRK?
No, the GRK phosphorylates the GPCR. Yeah.
Okay.
So it's actually when your GPCR is activated, then it activates the GRK, but that phosphorylates the GPCR. Does that make sense?
Yes.
So yes and yes and no. And yes. No. Okay. So what is the fate of the internalized GPCR arrestin complexes? Where do they go? MVB, so I heard MVB maybe. Where else might they go? Huh? Might they be recycled? They might be recycled. Where else might they go? Lysosome. That'd be another place. So I think all three of those are possibilities. And they might all happen depending on the receptor. Okay. Okay, so let's move on. Let's move away from GPCRs for a moment. Let's talk about signaling through enzyme-linked or coupled cell surface receptors. Okay, so there are 6 classes. I'm really going to, well, there are 6 classes. Do not memorize these. You can think of them as 2 classes. One would be enzyme-linked, and the other would be enzyme-associated. Enzyme-linked would mean there's a kinase domain somewhere in the receptor, cytoplasmic tail, and enzyme-associated would mean that they bind to a kinase. So receptor tyrosine kinases are enzyme-linked. Tyrosine kinase-associated receptors means a tyrosine kinase is associated with the receptor. A tyrosine kinase means the kinase phosphorylates a tyrosine. Okay? It's like a new language, right? I feel like I'm teaching Spanish, but in English. And then there's these other ones, receptor serine, greenine kinases. So again, these are going to phosphorylate serines and threonines, but they're receptors with kinase domains that actually phosphorylate the serines and threonines. And so it can be as complicated as you want, but really just put it in their 2 basic categories. Yeah?
You said enzyme linked, they have a kinase domain in their receptor,
Yeah.
and the other one they
And
just bind to
associated they
kinase.
bind to. Yeah. So those are your 2 main categories. Yes.
So do you want us to know which ones are part of which category?
No, that's not useful. Just understand the basic concepts. The basic concept being either it's going to bind to or it's going to have its own kinase activity. Yeah. Okay. So receptor tyrosine kinases kinase subfamilies have common domains, which makes sense because they're kind of all related. You're going to have these kinase domains, and that's shown in red. So each receptor, so remember, receptors have an extracellular domain, a transmembrane domain, and a cytoplasmic tail, right? And so the big thing is that they've got these kinase domains down in the cytoplasmic tail. It's not another protein, it's a domain within that one protein, okay? So when ligand binds, it activates the intracellular kinase domain by bringing them close together and inducing receptor cross-linking. So you actually have, so here you can see 3 examples of 3 kinds of ligands. Here's a growth factor that's secreted by some other cell. Here's, what is this? Some other secreted molecule. And here they're actually showing another transmembrane protein involved in the binding. Here it's actually another cell. This is cell-cell communication where you have a signaling cell and a target cell. And so the signaling cell has some kind of receptor that's going to bind to receptors in the target cell. But in all cases, these are all receptors that have kinase domains, and they're showing them like little bumps with fireworks coming out, that will phosphorylate themselves or phosphorylate each other. So it's the ligand binding that causes the receptor change. So there's usually dimer, and they might be further apart, and then the ligand binding makes them come close together, and they're brought in close proximity so they can phosphorylate each other, and you get this phosphorylation on the cytoplasmic tail. Okay, great. So I'm going to give you guys a quick practice problem to think about this, and then we'll talk more. So you're studying a new receptor, and you believe it's involved in regulating expression of genes associated with cytokine secretion in the presence of gram-negative bacteria. Design an experiment to determine whether a receptor functions in this signaling pathway. So what I want you to start thinking about here is how receptors are activated, what things need to happen on the extracellular side, on the intracellular side, how different mutations can affect the activity of those receptors, and how doing those things will affect gene expression, okay? So I'm going to give you guys a few minutes, and I'm going to go around this way. And when I come back around, we'll discuss, okay? Does anyone need clarification on the question, or are you good? Okay. Good. Go. Talk to your friends. Have fun. That's so confusing. You're so easy.
I don't know where to start.
Okay, so you're trying to figure out a receptor
Ligaments associated with that.
Well, you know
And then maybe I'm just not sure.
Okay, I wanna make a quick clarification. Quick clarification, because some of the questions I got made me realize that people aren't completely understanding the question. So just to clarify, you have a tissue culture plate with cells in it, and you add the bacteria, and the way you know signaling is turned on is if your cytokine's being secreted. And you're trying to figure out, so that's kind of like, did gene expression turn on or not, right? So, and you're trying to figure out, well, is your receptor, whatever receptor in question, responsible for this? And what kind of manipulations can you make to test whether or not your receptor is involved in the response to these bacteria, with the readout being cytokine secretion, okay? So that's me making you apply things, because you already know signaling is something outside the cell causing some signaling pathway to happen, and that's going to affect gene expression. Gene expression means proteins being made, right? So if you need another minute to clarify your thoughts, I'll give it to you. Does that make sense?
Yeah, I mean, just kind of thinking, you could inhibit a kinase in some way, if you knew what was supposed to be expressed out of the pathway. Yeah, it's just kind of test to see whether they get
Okay, guys. I gave you too much time. Okay. So what do you think? What do you think? Any thoughts on what you would do? How would you know? So you've got your cells and you've added your bacteria, and you want to see if your receptor, whatever one you're interested in working on. Remember, there's thousands of receptors on a cell. You don't know which one is responsible for signaling that was resulting in turning on cytokines, right? Turning on gene expression that results in production of cytokines and secretion of cytokines. So how do you figure out if your receptor is responsible? Come on, I know someone else. Yeah.
Yeah, I'm gonna block your receptor, like block your receptor signal.
Okay.
Block the, what is that, cytosolic?
Block the cytoplasmic tail.
And if it's still secreted, then it doesn't have any effect.
So block the cytoplasmic tail. If your cytokines are still secreted, then then your receptor is not involved. How would you block your cytoplasm? What could you do to block? So block signaling. How?
Branding the receptor, like the phosphorylated and the racemab.
Okay. So if we were talking about GPCRs, somehow induce endocytosis, but we're talking about RTKs. So let's talk about receptor tyrosine kinases. So the arrestin is specific to GPCRs. Now you could also somehow induce endocytosis of your receptor in C, but I want you to use this information that I'm giving you about these kinase domains. Yeah. So the question is about the function of kinase domains and how that affects activity. Okay, that's what I'm hearing. So we'll go further into that. But kinase domains phosphorylate and often create docking sites for other proteins that are going to participate in taking the signal all the way to the nucleus. Yes?
Could you somehow take all the phosphate out of the medium?
Okay, so remove the phosphate from the media. That would be good, except for that would affect every phosphorylation function in the cell, but you're close. You're close. Yes?
Could you just mutate the kinase domain?
Can you mutate the kinase domains? Why not mutate the kinase domains? That's more specific, because that's what you're wanting, right? So mutate the kinase domains. Yep. Okay, mutate the kinase domains, then you can't get phosphorylation, you can't recruit other proteins, and you can't get signaling. And so if you still get cytokine secretion, then your receptor's not involved. But if cytokine secretion gets turned off, then you know your receptor is involved. So that was collaborative. We pieced it all together with all the different ideas. Okay. Does anyone have questions about that practice problem? No. Is that because you have no idea what I'm talking about, or is it because it was so simple you couldn't be bothered? Who said yeah? Okay, come see me if you need clarification. Okay. So autophosphorylation of receptor cytoplasmic tails activates other kinases and creates docking sites for other intracellular signaling proteins, forming a signaling complex. So all that phosphorylation creates these binding sites. And these are tyrosine kinases, so they're phosphorylating tyrosines. So anything that has a domain that will bind to a phosphotyrosine that's close by has that ability to bind. So things with SH2 domains, remember we talked about SH2 domains, they like to bind to phosphotyrosine. So then they can bind. And that forms a signaling complex which helps to relay the signal to the nucleus. So in here, what's the role of phosphorylation in signaling? Is it activating or repressing? activating. Now, could it be repressing? I guess it depends, right? It depends on what's being recruited. So here I'm telling you, you're recruiting all these proteins that help to form the signaling complex that activates gene expression in the end. But let's say it recruited an inhibitor. So your answer should probably be, well, it depends. No, there's no question. I don't know. The question was, what's the role of phosphorylation in signaling? And I got, it's activating, but it's activating based on what I told you. But if I told you that phosphorylation recruited inhibitors, which is weird in this case, but if I told you phosphorylation recruited inhibitors, then it would be inactivating. So you should always say to yourself, well, what's that phosphorylation regulating? What's it recruiting? Okay? And so one way to inhibit signaling through this receptor is to somehow inhibit phosphorylation or recruit some inhibiting complex. So, so I was just saying there's all this phosphorylation on tyrosines, right? So you get binding of a signaling molecule, and the receptors come together, and the kinase domain is phosphorylated all over the place, and all these tyrosines are phosphorylated. It looks like this. And that allows proteins that have SH2 domains to come and bind. And so this is just showing that, this is kind of showing the binding site on a protein. So, so it's all about changing shape. So you know how I told you about how phosphorylation changes the shape of a protein? So you might normally, in a protein, have this binding site, this teeny little thing, and that's perfect site for phosphotyrosine. And if that's not there, then maybe you'd have another space that would bind something else somewhere else. And so it's about regulating the shape of the proteins and regulating the interaction, and that will have effect on the signaling process. So you can have these proteins with these SH2 domains, and they might interact with phosphotyrosines on activated receptors. They might increase or decrease signaling, and it all depends on what's being recruited. So I don't want you to ever think that phosphorylation is activating, period. That's just how it is. It's really about recruiting proteins, and whatever protein's recruited is going to have whatever effect. So if I give you a question on an exam, I'll give you the information that's needed so you can decide what the effect will be. Okay, so let's talk about another GTP binding protein, RAS. So RAS is another molecular switch. So what can you tell me about RAS in terms of regulating RAS activity? Just based on prior knowledge. Yes. It depends on GTP. So what do you mean? So is it on or off when GTP is bound? On. Right. And what turns RAS on? Some kind of GEF. And what turns RAS off? Some kind of GEF. Right. So exactly. So this is how I want you to start approaching cell biology. You've got all this prior knowledge, and there are themes that keep coming up over and over again. And just apply what you already know, and you might be right. You guys were right. So, what is the effect of a mutated RAS that? Well, let me take this back. Okay, so RAS, I tell you it's a molecular switch. It's turned on, it's turned off based on GTP. Okay, we're good. What it does is it relays the signal from the cell surface to different signaling pathways. So you have your receptor that's been bound by a signaling molecule, and you had all this magical phosphorylation, and proteins are recruited, and somehow one of those proteins is a RAS GEF, which is going to activate RAS and put a GTP on it, and then RAS is going to signal and activate different pathways. Sometimes RAS can activate multiple pathways. So if you have a mutation in RAS where it stays in the GTP bound conformation, what's the effect? What's the effect on signaling? Yep.
The signal isn't gonna go away and it stayed on.
Right, signaling's not going to go away. It's not so remember, signaling needs to be regulated. It's only usually turned on when a ligand binds or there's some kind of signal coming from somewhere to activate the whole process. But if you have RAS that's stuck in a GTP bound state, it's going to be stuck on signaling multiple pathways with zero regulation. Is that good? So what kinds of mutations do you think might be associated with cancer? Ones that are involved in keeping it on, or ones that are involved in keeping RAS off? Ooh, I got on and off. Okay, why? Who says on and is prepared to defend their opinion? Huh? Okay.
Well, cancer cells need to divide, so if you keep RAS on and the signal is gonna keep coming, so the cells can divide. If you turn it off, then the cancer cells don't get.
Okay. So the question, so you're right. So what Rebecca is saying is cancer cells like to divide, and so if you have RAS and it's stuck on, and that signal, and what you needed to know is whether that signaling pathway is associated with cell growth. But assuming it is, which many are, if it's stuck on, you're going to have constant cell growth. Now, who says off? Yeah.
I said off, but now I agree with her.
So easily convinced. Well, yep.
I just see how it rises all right there. Because if whatever's being made, whatever process of RAS is to stop or kill the actual cancer cells or whatever the elements are, if you're stopping that process, it's going to be built up.
Okay, so you're saying, so are you talking about pathways that might signal apoptosis? And if you get stuck off, then you're not gonna go through apoptosis. So I think that you can go either way there because there are signaling pathways that induce apoptosis. So I think there's both. And so whenever I tell you there's a mutation, you're gonna want to ask me more detail, right? Yeah, good. Okay, so like what we all just said, RAS activity is activated through recruitment of a RAS gap to the RTK signaling complex. So remember we had our RTK and the signaling molecule bound and there was all this phosphorylation, right? And then that creates all these different binding sites. And one of the binding sites recruited this adapter protein called GRB2, don't worry about that detail, which then recruited a RAS gap. And so RAS is a membrane-bound protein. It's hanging out, bound to the membrane, it's stuck there, but it's in its off state. It's GDP bound. But signaling, what it does is it recruits RAS gap. And then what happens? GDP is kicked off, GTP is allowed to come and bind, and then you get active RAS, and then you get transmission of the signal to multiple pathways. Okay? So RAS is tightly regulated, right? So when you're thinking about this, I want you to ask yourself, how is RAS's activity regulated? How is that related to signaling? I just told you, so I won't make you answer now, but I definitely want you to be thinking about that when you're studying. Why isn't RAS active all the time? In a normal non-cancer cell, why isn't it always on? No one? Well, you need recruitment of your RAS gap to turn it on. So it's going to normally be off. And then the other thing you need to know is that RAS has a well, there are gaps that associate with RAS, and then RAS also has its own GTPase activity, which means it has its ability to hydrolyze its own GTP. So that prevents it from being stuck in the on position, or the on position, in the on conformation. Okay, and that's a good thing, right? Yes.
Is that GTPase activity as like an override system, so it remains on and it can tell itself to turn off, or vice versa?
So the question is that GTPase activity like an override system so it can turn itself off if it gets stuck on? I think you can kind of think of it that way. I think there are multiple ways of regulating proteins that are involved in signaling pathways. And since RAS is so important in activation of so many pathways, there needs to be many ways to make sure you can turn it off. So that's why there are multiple gaps that are going to be recruited under different conditions. And then also you have this, I think it's a fairly slow but efficient hydrolysis of its own GTP. So you can think of it that way. Yeah. Any other questions? Thoughts, comments? Okay, so here's RAS signaling in cancer. So that's a lot going on there, so we won't make you memorize it just yet. No, I won't ever make you memorize it. But so just goes to show you how many different pathways. Look at all those different receptors and all those different signaling molecules that are regulated by RAS. So you can imagine that mutations in RAS are gonna be very much involved in cancer, and they are. And the different pathways that RAS is involved in are involved in cell proliferation, so cell growth, cell survival, differentiation, cell cycle control, another thing, cell motility, and tumorigenesis, and tumor-associated RAS, so RAS that's in tumor cells, is often constitutively active, so it's always on because of point mutations in the gene. So you can guess where those mutations might be, right? Where do you think mutations in RAS might be if you're a cancer cell? Remember, proteins have different domains that do different things, and so what you know already, you can make certain assumptions on what RAS might do based on what I told you, right? RAS is a GTP binding protein, it needs to bind to this, it needs to bind to that, its activity is regulated by that and this. Come on, guys. Anyone? What kinds of mutations do you think you might find? Like what part of the protein do you think there might be a mutation in? You know what? Don't tell me. Don't tell me. I don't want to hear it because I think I have a practice problem. Yeah, I have a practice problem coming right up. So hold that thought. Hold that thought. Oh, it's going to come back. It's going to come back. Okay. Okay. So the RAS MAP kinase signaling pathway continues to relay the signal downstream by phosphorylating gene regulatory proteins and activating other kinases. So here's your active RAS, and it goes and activates a kinase, which phosphorylates and activates another kinase, which phosphorylates and activates. It's like that, right? It's a whole lot of high-fiving between kinases. And then you'll get phosphorylation of proteins that change the protein's activity and maybe affect something that way, or maybe affect gene expression by allowing a protein to dimerize, to go to the nucleus, or degrading an inhibitor, or you can start to think about all these things that phosphorylation can do. You know what we should do? Oh, I have a great idea. We should do like a make up your own signaling pathway, but it has to all be logical based on what I taught you. I'm thinking. Okay, so let's do the practice problem first while I come up with a fun thing to do. So you're studying pancreatic cancer. That's a good one to study because that's a particularly bad cancer to have. So since RAS is frequently mutated in cancer, You decide to sequence the RAS gene in your cancer cells. So you're in a lab, you have these pancreatic cancer cells, and you're like, well, I don't know what's wrong with these cells, but let's check RAS. So you sequence the gene and you're checking for different mutations. You find a common mutation in your tumor samples that results in a G to V mutation at position 12. It's a mutation that results in amino acid change. You look in the literature because you ask yourself, well, has this mutation been described before? And you find that this mutation falls into a gap binding domain. Hypothesize the effect of this mutation on RAS protein function and the effect on the signaling pathway. Is this an activating or inhibiting mutation? Is this mutation contributing to development of cancer? And design an experiment to test your hypothesis. So I'm going to give you a few minutes to discuss with your friends, and we will see. And so this is just showing your RAS protein sequence, and there's these GTP binding domains that are in red, but I already tell you here that it's in a gap binding domain. Okay.
Rather than it's disallowing it for GTP to be cycled. That means that you hydrolyze by testing for the levels of RAS, cancer cell versus
No, right?
I just don't want to
And see,
Okay, guys, let's see, what did you guys come up with? So, what do you think that, so we found a mutation that seems to fall in the gap binding domain somewhere. So what do you think the effect is on the RAS protein's function and the effect on the signaling pathway? So what do you think it'll do to RAS? Yes?
Want to keep it in its active state since the gas is not taken off the oxygen?
Right, so will it keep it in its active state because GTP is not being hydrolyzed? Okay, right? So that's one thing, but then remember there's this kind of slow GTP hydrolysis that happens because there's a GTPase domain, but it's not as effective, so it will be active for longer. So you're on the right path. So what's the effect on signaling? Remember I told you it's involved in all these different pathways, right? So if you're stuck on a lot of signaling, right? So do you think this mutation might be contributing to the oncogenesis, the development of cancer? Yes. Right. So what would you do to test your hypothesis? So you think that maybe it's keeping it on? Yep.
No, I have a question.
Yep.
So this is an inhibiting mutation because
It's
it's inhibiting GAP.
inhibiting. So it's inhibiting gap interaction.
So it's an inhibiting mutation.
I don't know. It's inhibiting gap interaction. How does that affect RAS? Right. So it's inhibiting the inhibitor. Right. So in a way, it's inhibiting interaction with gaps, which should turn off your RAS. So you're stuck on.
So is this an activating mutation?
Is this an activating mutation? I would call it an activating mutation. What do you guys say? Activating?
Yeah.
Yeah. If we have consensus. Yeah.
Can you mutate your mutation?
Can you mutate your mutation? You can do whatever you want. Yeah. You can
Mutate
mutate.
your mutation so you can activate GAP, and then that way you definitely know that is GAP being inhibited, and then you left on.
So you would mutate your mutation and change it back to what it was and see what would you look for? What would be your readout?
If you still have uncontrollable growth.
To look at the effect on cell growth. Okay. I'll go with that, yep.
So could you mutate the GTP domain on RAS?
So it
Mutate the GTP ACE or GTP binding.
Does RAS have a GTP domain?
RAS has a GTP binding domain, RAS has a GTP ACE domain.
So can you mutate the GTP binding domain?
Okay, so it can't bind GTP?
And then see if you still have signals?
Right, but what do you think will happen? Okay, so I'm hearing mutate the GTP binding domain, see if you stop signaling. You should know the answer to that though, right? If you don't have GTP, will it be on? No. So it doesn't answer your question though, it's a good way to kind of test proof of principle. I think looking at cell growth is a good way to go because that's what cancer cells do. Yep.
Can we somehow increase the speed of the RAS
Try to increase the speed of the GTPase. Well, so that kind of goes with what I was hearing over there is mutate it back, right? Mutate the gap binding domain back, and that in a way would increase the efficiency of GTP hydrolysis, and then look at gene expression or cell growth.
Can you mutate a normal protein, like the normal cell, watch it and see where it goes?
Yeah, that's another thing you can do. You can mutate your happy, healthy RAS and see into the same mutation and see how that non-cancer cell begins to grow, or how it affects gene expression in the non-cancer cell. Sure. Yes.
Can you take another RAS protein sequence, make a mutation, not at the protein cell, but somewhere else, and check it that will line will have so uncontrolled cell.
So can you take another RAS protein and mutate it somewhere else? Where?
Another protein.
Another domain. We have to be specific when you're making use because you need to mutate with purpose. Mutate with purpose. We're going to make T-shirts. Okay, mutate with purpose. Yeah, I think you need to think through. You're going somewhere, but you need to think a little more. Anyone else? And then I'll move on. Okay, lots of good ideas. Lots of good ideas. Yes.
I have a question. We're like learning about this like similar stuff in molecular now. So is this would be an example of a driver mutation?
A driver mutation. So I want you to remember, I teach molecular in a different class. What would a driver mutation do? What does he mean? Because I don't teach driver mutations.
Well, we had like a homework assignment about it. So a driver mutation is a mutation.
That drives cancer?
Yeah.
Sure. Well, so then there's like that mutator phenotype. Is that what you're talking about? Kind of? Like there's like the mutator phenotype where like you get mutations that then encourage more mutations, which then it like messes with the DNA repair machinery, which encourages more mutation, and that kind of drives cancer development. So maybe?
Like the one I did in my homework assignment was a mutation in the gene that creates growth factor receptors. So
EGF?
Yeah. So that one. So that was like the example that I was
Okay.
using for it.
So in that case, I think if a driver mutation is anything that drives the development of cancer, then I would say yes.
Okay.
Yeah. It's funny, someone came to me the other day for help in Molbio. And I was like, but I don't teach, I'm not Dr. Weldon. If you take my class, I can give you help. Because she was asking me all sorts of things. I was like, Okay. Okay, we did that. Nope. Yes. Okay, so MAP kinases, so we're going to move on. So we're going to talk about RAS. Let's remember where we are, right? Let's go back for a minute. So remember we had signaling, receptor, adapter protein, RAS. GEF activates RAS, which then activates a bunch of kinases, right? Kinase. Phosphorylates things, activates gene expression, or maybe changes some protein activity. So these kinases that I'm talking about, MAP kinases, there's usually 3 of them, right? MAP kinase, MAP kinase, and MAP kinase. It's very, very special the way they name those. MAP kinases, they're involved in so many different signaling pathways. There's so much chatting here. Do you guys have something awesome? I love awesomeness. No? Okay. I get so distracted so who gets distracted when they hear other people talking? I do. It's so bad. What I need to do is drink a cup of coffee before I teach. MAP kinases are involved in so many different signaling pathways. So you need to think to yourself, wow, they're involved in so many signaling pathways, and the cell is a very crowded place. What's preventing them from activating all sorts of crazy things? But really what they need to do is MAP kinase needs to activate MAP kinase, which needs to activate MAP kinase, which needs to then activate the next thing, right? But if you're not careful, then this mating factor might activate glycerol synthesis. We don't want that. We want it to activate the mating response. So you want to keep the signaling complexes together by using these scaffold proteins. And scaffold proteins act just like scaffolding on a building, I guess. Do they? I feel like scaffolding on a building is where people stand to do things. Okay, so that's kind of where the kinases stand to do things, right? But it keeps them all together. I don't know that a scaffolding on a building keeps the people all together because they have freedom to leave and so does the protein. Anyways, I digress. So scaffolding helps to keep the signaling pathway signaling the direction it needs to go so that kinases activate each other and not go and activate another pathway by accident. Okay. So here's the MAPK signaling pathway, and you can see, again, lots of receptors, tons of MAP kinases involved signaling, and it's important to keep them together so the right pathway is activated. And that's why these scaffold proteins come into play. So a mutation in some kind of scaffold protein, as you could guess, would be completely disastrous to the cell. Are you asking me if you could memorize this? Oh. Okay. How are we doing? Is everyone doing good? Are you guys still awake? Uh-oh, I see Donna. Okay. Okay. So another thing that can happen is so you can get plasma membrane bound PI3 kinase, which will phosphorylate inositol phospholipids. And that creates docking sites for intracellular signaling proteins. Okay? So this is a whole nother way of signaling that starts with an RTK, right? So we talked about RAS and what happens there, okay? And this is, notice no RAS here. This is another thing that can be activated. So I don't want you to get stuck with like, you know, this signals through this, signals through this. It's actually, there's a lot of different mechanisms for each class of receptors. So you can get this PI3 kinase, which will phosphorylate the PIP2, and that, again, what it's doing is creating docking sites for recruitment of different proteins that then will phosphorylate BAD, which is involved in keeping inhibitor of apoptosis inactive. And then when that's phosphorylated, then the apoptosis inhibitor becomes active and can inhibit apoptosis. And this all has to do with these phospholipids that get phosphorylated and then recruit proteins to the membrane and then activate other proteins. And it's all similar in the sense that you're phosphorylating things, creating docking sites, recruiting other things, activating kinases, and then you get this final phosphorylation that's going to somehow affect the protein. Notice here you don't necessarily have a transcription factor going to the nucleus, right? I use that as my most common example of signaling, but here you're actually activating an inhibitor of apoptosis. Ooh. You raising your hand? Okay, how's it going here? Can I take the temperature of the class? Are we on fire? Yes.
So this doesn't happen if you don't have inactive inhibitory protein phosphorylated, then you get
Right. Right. And sometimes you want apoptosis, right? But if this is dysregulated and this gets phosphorylated and you have this dysregulation activation of an apoptosis inhibitor, right? is, I know, she just was like, I'm on fire. Okay. If you inhibit apoptosis, is that good for the cancer cell? Well, I guess it depends. It's good for the cancer cell, it's a very successful cancer cell. Not good for the organism, no. Okay, I'm gonna let you guys go with this and you can think about this and we will continue.